Thank you for coming to this lab roots talk. I'm David Danko. I'm the Chief Technology Officer of Biotia, a New York City-based startup focusing on infectious disease diagnostics. And today I'm going to talk about pandemic early warning systems. And what I'm going to hope to convey in this talk is why pandemic early warning systems are necessary and important, what sort of data types can go into such a system, and give you a few motivating examples both of where a pandemic early warning system could be helpful and how we could build such a system. The COVID-19 pandemic revealed that global health, social systems, and economies are exceedingly fragile and increasingly interconnected and interdependent world. In the last century, in the 20th century, public health made extraordinary progress against infectious disease. At the beginning of the century, sanitation systems effectively wiped out urban miasma like cholera and typhoid in cities. And in the middle of the century, vaccine programs alleviated the burden of childhood illnesses such as polio and measles in much of the Western world. This extraordinary progress has been stress tested by a single pathogen, SARS-CoV-2. Had there been an effective tracking system at the start of the pandemic, public health officials would have been able to make more informed policy decisions, potentially leading to different or mitigated results. Absent the maintenance of robust and international frameworks for infectious disease monitoring and data sharing, localized biological risk will continue to transform into global shocks and pandemics. Climate and environmental stressors will intensify these shocks and make them more likely, while globalization will make them universal. The risk of climate change in the spread of pandemics is particularly prevalent. Current estimates indicate that about half of all infectious disease could be made worse by climate change. This is largely due to increased habitation and increased environments that are conducive to mosquitoes and other vector populations. Where there are more mosquitoes, there's more risk of mosquito-borne disease. And while the social and societal effects of pandemics are highly complicated, we believe that the underlying factors that lead to a pandemic are fundamentally predictable and can be figured out. What we envision is a system, a platform that is open source, that is global, and is focused on community driven, that eventually creates quantitative factors that lead to effective risk maps and prediction for pandemic and epidemic outbreaks, leading eventually to improved public health outcomes. The animation that you're seeing on the slide here shows the biomass degradation in an area over a period of approximately 24 years. It's built from Earth observation technologies, including LiDAR and satellite data, and measures the total biomass in a region. What you can see is that the biomass degradation for this region begins near roads and spreads out from there. And this degradation of habitat leads to the potential for animals to become vectors and for increased risk of zoonotic disease. So of course, this is a biology-focused talk, and we'll be talking more about how biology and genomics data is a critical part of a pandemic early warning system, but we don't want to become myopic on one data set type. Our belief is that a pandemic early warning system relies on a lot of different data types. Biology is certainly one of them, but so is climate and weather data, social dynamics, infrastructure data, maps, public health data, and epidemiological data, and social determinants of health, all of which are going to be relevant to making effective models of pandemic warnings and the factors that lead to pandemics. Really, what we're trying to do is create a modern logistics of data that puts this data in the hands of local teams, community-driven teams, that are going to be best equipped to understand their particular environment and their particular systems that they are expert in. By combining this logistics of data, we will be able to break data silos and democratize AI tools in a way that's going to give people access to some of the most high-powered algorithms, satellite data, imaging data out there to create really effective predictions, which will answer some big questions. And the kind of questions that we're looking to answer are things like, what is the current level of habitat loss for a given region? Where is this occurring in relation to plantations mill? Based on past practices, can we predict or model what may happen if these trends continue? Our belief is that through the development of thoughtfully designed technological platforms, we can build global scale precision disease and detection response systems. These will reduce exposure to systemic shocks, accelerate science-informed public health policy, and deliver reliable healthcare and economic opportunities as part of an intrinsic part of a global sustainable development agenda. One reasonable question here is why we believe that this is possible now. There have been significant advances in technology, particularly in the last 5 years, but over the last 30 or 40 years, that make this kind of pandemic prediction possible in a way that it wasn't before. Really, there are kind of three main factors that drive this. the emergence of large, complex data sets, which span hundreds of thousands to billions of human interactions, as well as sequencing data, which allows us to probe biology in a novel, incredibly deep way. High performance coding, high performance computing and algorithms, which allow us to process this data in a way before. Particularly, this is related to the performance of graphics processing units and the incredible increases there, as well as machine learning algorithms based off of those processors that provide insights like protein folding and machine vision, which were essentially science fiction 10 or 15 years ago. And the third factor that we see is the growth of global talent pools. What we like to say is that talent is global, but opportunity is not. There are smart people everywhere. And what we want to do with this platform is give those smart people the tools to improve the public health of their environment and make changes in their own realm. Of course, there are already some organizations doing this, particularly the WHO World Health Hub and the Africa CDC have missions associated with this already and are building out programs around pandemic early warning with many of these ideals of community-driven modeling and social determinants of health in mind. So the key takeaways that I want to leave here is that there is an urgent need for an ongoing pathogen surveillance and global early warning system, particularly due to climate change. Such a system needs to incorporate many different data types, not just genomics and climate change data, and that building such a system is going to require us to look at data in a new way. This data has to be community driven. It has to respect the data sovereignty of people putting this data. And we need to democratize a lot of the tools and data around this. So with that in mind, I want to switch gears a little bit and start to talk about the role of genomics and the need for other data streams in this and to sort of get into the meat of how we can start to build such a system. So probably this slide is going to be a little bit of review for many of the people at Lab Roots of, why is genomics an important part of this biological database, and particularly why is sequencing such an important part of this? Well, review for a lot of the biologists out there, but all living organisms, unless you count prions as living, contain DNA or RNA. And that genetic material lets us map pathogens. Now, really what I want to say here and the point I want to make is that sequencing, particularly sort of metagenomic, environmental, metatranscriptomic type sequencing, lets us perform biology in a massively multiplexed way. So historically, very historically, you would kind of go sort of one organism at a time in a sample. You would try to culture a particular bacterium, or you would sequence a couple of genes via PCR, or maybe a little bit larger panel with something like a microarray. But by definition, you were just looking for one organism or a few organisms, and you were doing it in a single way. Sequencing kind of reverses this. So now you can perform a single assay that looks for pretty much everything at once. And using that, we can do a much better, more complete picture of what the biology is in a given sample. Now, in particular, this use of genomics was very important during the pandemic. And I'm going to show a little bit of a video here on the next slide of how the strain dynamics of SARS-CoV-2 played out during the pandemic, which of course is a genetics-based assay for how that works. So I'll switch to that video now. So what we could see in a video like that is that over time, the strain dynamics of SARS-CoV-2 changed. We were able to map out where strains were going, and we could make these patterns showing where strains were transferred from, where they were transferred to. And this was all built on sort of these ad hoc systems during the pandemic, particularly the GSAID platform stepped up to move away from just a flu and the diseases it typically did into also supporting genomics data. The visualizations there you saw were all from NextStrain. A lot of this was often put together last minute, was done by these sort of heroic efforts rather than a systematic effort to be able to map and track pathogens that could work for a wide variety of in an emergency setting. So what kinds of things can we actually pull out of a metagenomic data set that are going to be useful for a pandemic early warning? So of course, any kind of metagenomic or metatranscriptomic data set is going to let us find viruses, bacteria, pathogens, fungi, certain extent. But we can get more than that. When we take a metagenomic sample from a particular area or a particular region, we can find antimicrobial research. You can find evidence of host DNA or RNA that are going to help us map out what kind of organisms are potential reservoirs for diseases, what are hosts. We're also able to find important biosynthetic things, biosynthetic gene clusters, CRISPR regions. We'll be able to map out strain evolution, and we can find novel viruses as well as novel other potential pathogens there, helping us to get ahead of the next threat before it even happens. Genome, and particularly data sources like metagenomics, viral sequencing, bacterial sequencing, are an incredibly informative data source for pandemic early warning systems. It doesn't necessarily tell us the full story here. In particular, we need to look at climate data and Earth systems data as an example of how these things do. To give a key example, mosquitoes, of course, are bred in certain environmental conditions. They require groundwater to lay their eggs. They are most active at particular temperatures. So predicting disease doesn't rely on just identifying the presence of mosquitoes or the presence of particular viruses, which can be done through biological assays, qPCR assays, sequencing assays, of course, but also by identifying regions where mosquitoes are likely to be breeding and are likely to be spreading these diseases. And really what we envision here is a system where these very high resolution but relatively expensive sequencing and biological samples are used to map out and to label satellite data, which is relatively abundant, relatively high resolution, and build these integrated deep maps where you can have real biological samples informing sort of predictions based off of the satellite data. And of course, social determinants of health, other public health data sets that will then inform also those predictions. And in particular, why this is important is because climate change, as we mentioned previously, is massively increasing the risk of zoonosis. This little animal that you see here, this is a rat that is common in the sort of the Americas and is well known for spreading hantavirus. And the reason I'm showing it here is to emphasize an example of how climate change can affect the kind of spread of a disease here, because hantavirus, largely transmitted by rodents. Rodents, particularly this rodent, are confined to different areas based off of deforestation, expansion of their range through improved temperature, has caused a massive increase in the rate of things like hantavirus and other zoonotic diseases as borne by the increased range of their hosts. And through mapping that, we'll be able to form a better picture of where this stuff is and how we can effectively prevent it. So the key takeaways I want to give here is that while genomics provides powerful insight into infectious disease dynamics, along with public health data, it basically forms the cornerstone of any biological and pandemic early warning system. We will benefit from incorporating varied data streams, including Earth observation data, climate data, social determinants of health, that are going to help us build this more 360, more effective picture of what's going on in the world. So having talked about why we should build a pandemic early warning system, and then a little bit about the kind of data sources that go into a pandemic early warning system, now I want to talk about a particular motivating example for how such a system would work. And I'm going to use the example of biosurveillance of mosquitoes in Brazil and some of the diseases there. So the first motivating example I want to talk about here is Zika in Brazil. Many of you will, of course, heard about Zika in Brazil in connection with the 2016 Rio Olympics. But what might not be well known is that Brazil actually didn't have Zika or any significant Zika presence before about 2013. We don't know exactly how Zika was introduced in Brazil, but it was likely in connection with a couple of mass sporting events. And this had a tremendous impact on Brazil's population. About 1,700 newborns were born with congenital Zika syndrome. Of course, there was some panic around the 2016 Olympics as people were concerned about going and getting Zika. And just, generally a very bad outcome, as one might expect from the introduction of a dangerous tropical disease for the first time. Now, why Zika is part of the motivating example here is that Zika was sort of a prime candidate for where a global coordinated early warning system potentially could have helped improve the response here. That's for a couple of reasons. Zika was a known virus, so sampling for Zika would have identified potential sequences. If it had a broad arbovirus panel or metatranscriptomic sequencing, would have been able to pick up Zika as both a virus and a dangerous human pathogen. And then the mosquitoes that transmit Zika in Brazil, Aedes aegypti, were well known. And of course, the habitats where they could exist and the temperature range where they're active was also something that we could predict with climate and meteorological data. So in this case, Brazil's public health response did respond to Zika quite quickly. Of course, Brazil has a lot of experience dealing with tropical diseases. So while having a new disease was difficult, they did respond to this. It's possible that with an early warning system, this response could have been mitigated even further and improved the situation for Brazil. Broadly, for pandemic early warning and epidemic early warning, mosquito-borne diseases and arboviruses more broadly are a really important thing to think about here. About 17% of all infectious diseases are vector-borne, and these cause about 700,000 deaths annually. Malaria is probably the best known of these diseases, but also things like dengue, chikungunya, Zika, and yellow fever are all really critical issues, particularly in sort of the tropical belt of the world. And the increase of climate change and the increased land use due to increasing population and deforestation, that kind of thing, are massively increasing the rates at which vector-borne diseases can improve. In particular, there are a few different maps and estimates showing that large parts of the southern US could be susceptible to these kind of vector-borne diseases within a few years. Brazil, in particular, has also seen a significant climate change-based expansion of some of these diseases. Yellow fever, now common arbovirus in Brazil, was once sort of confined to a particular region of the Amazon. And due to deforestation, due to climate change, due to mosquitoes sort of being able to hop on trucks and move around, then this has expanded increasingly throughout Brazil and now is responsible for sort of 2,000 cases and 700 deaths a year about. So a significant problem with these yellow fever in Brazil. And a pandemic early warning system could certainly have predicted some of the habitat change and some of the effects there, but could also help in the future by showing where vaccines could be effectively targeted, where populations are at most at risk, and deploy these scarce resources of yellow fever vaccines to areas and hopefully mitigate some of the negative impact there. And I want to highlight now one project. So all of this data has been from a field crews, a group in field crews of Brazil. And I want to highlight one particular project this group is doing that is sort of centered on identifying mosquito and early warning system. In a city in Brazil, in Foz do Iguaçu, they have about 85,000 trap inspections, mosquito traps throughout the city. For 5 years now, they've been collecting mosquitoes in all of these traps, mapping out where the mosquitoes occur, running PCR assays on the mosquitoes to map disease, and relating public health dynamics and data. And this study has led to a really detailed picture of mosquito and arbovirus disease in Brazil, in this one city in Brazil, in Foz do Iguaçu. But it's inherently kind of a local picture. It demonstrates that we can, with the right data, map out this disease, make effective predictions, know when resources need to be deployed and where they need to be deployed to. But what we need to do is scale it up. We need to think bigger. We need to go from just an individual city to an individual country to a global prediction system that's going to be able to incorporate data from all these sources and build a broad, effective predictive model. I'll leave here the takeaways is that if we adopt this sort of philosophy around sharing data, around building these global systems, around broad testing, even for pathogens that are present in a particular country but could be introduced due to climatic reasons, we will have a much more effective global public health system. We'll be much better prepared for future shocks to that system, and we'll be able to distribute scarce resources such as vaccines and medication in a much more effective way where they're going to help save lives and help people. So now I want to talk about the last piece of this, which is how we can build some of the nuts and bolts so we can actually create this pandemic early warning, pandemic prediction system, how we can get out of it. We talked about pandemic warning, and I want to highlight that with the timelines that we're dealing with, it's not just enough to understand that pandemic is happening. We really want to be able to predict pandemics so that we can start to formulate a response or formulate plans of action even before these become crisis levels. This is because now things spread so fast due to globalization that it's not really possible to wait and still coordinate an effective response after something is already happening. So some of the data sources that a pandemic early warning, pandemic prediction system can use, we've talked about some of these in broad strokes, but I want to highlight some of the specifics here. Urban samples from urban environments, subways, environmental metagenomic samples, wastewater, air, maps of urban environments. These can give us this really high-depth picture of locations that affect a lot of cities. So things like this that you should pay attention to are the Metasub project, that Foz do Iguaçu study done by Rafael Freitas and his group at Field Crews, and a few other studies beside. In addition to these urban samples, we need to understand the broad sort of the countryside, the rural areas as well. To do that, we can do samples from watersheds. Target expeditions can seek things out. For example, one thing they do in Brazil is they go, and if there are sort of monkeys that die, they sample those monkeys to test for the possible presence of disease and vectors that could affect humans. And these efforts can effectively map risk in lightly inhabited areas and can also identify things that are potential zoonotic factors that could hop over humans. Mapping vectors directly, particularly mosquitoes, will also identify spreading disease and can coordinate response. So by taking mosquito samples, running either qPCR or sequencing-based assays on them, we can understand where disease is likely to spread, where it's going. And we can then coordinate that with satellite and Earth systems based imagery to identify high-risk areas where these mosquitoes are likely to go, areas where zoonotic diseases are happening, deforestation, that sort of thing, and then correlate that with health records that provide validation of spreading disease and predictions. With that data in hand, how do we actually identify potential epidemics as we happen? There are basically three tiers that we can look at here. First, ideally, what we would want to be able to do, and this is a little bit science fiction at that point, but it's also a little bit real, is to be able to simulate a particular pathogen. So you identify a particular sequence from somewhere, and you want to say, does this sequence present a health threat? Does it present something that we need to be worried about? So this is, not possible. You can't take a sequence and say this is definitely going to be a human pathogen, even though we've never seen a sequence before. But some pieces of it are there, some possibilities are there, which are important to pay attention to and important to continue to develop techniques on. Particularly if you see a sequence that's very similar to a known virus, that's of course a high risk. But also if you can simulate sort of a protein fold and find something that looks like a protein on a known virus or a dangerous mutation that's been identified in other places, we can make some steps towards being able to simulate and identify potential risk factors even before anyone has gotten sick or before the virus is spreading at all. The second sort of tier of identification is to identify expansion of organisms, particularly exponential expansion. So the hallmarks, virtually the definition of an epidemic or a pandemic, is the exponential increase and spread of a disease among a population. So in order to identify that as early as possible by monitoring sort of wastewater systems, air systems, whatever we can do at regular intervals, we can look for particular organisms or particular K-mers that are found exponentially more often. And as we track these time series, we can identify full organisms. We can sort of head off these events, potentially combine this in the simulation to improve our ability to respond quickly once cases do break out. The final piece of identifying a potential epidemic is actually identifying the first few people infected and to quickly understand what they're infected with. Presented with a novel case study, particularly things like a novel respiratory virus, it's critically important to find those first 10, those first 100 patients, see if we can find a consistent causative factor, a consistent pathogen causing these illnesses, and then coordinate clinical and public health responses accordingly. Now, overall, whenever we're talking about pandemic prediction and pandemic early warning, we have the motto where a case is too late. By the time you have a case, someone has already gotten sick. There's already been an adverse event. So as much as possible, you want to reduce cases. We want to reduce the reliance on people getting sick for us to have data. But that's not always possible. And using the first few people to get sick from a disease will at least prevent the vast majority of other people becoming ill and having an adverse outcome. To do this, one thing Biotia is working on is setting up what we call the pathogen watchtower. This is a set of about 12 sites all over the world where we work with local academic and research groups to set up sequencing studies, to do environmental or other similar assays that can feed into a global coordinated pandemic early warning system. As I said, there are about 12 groups here, but we want to highlight 3 different groups and what they're doing. In Chile, there's a group looking at bird droppings and looking at potential avian-borne viruses at avian migratory hotspots. So these are birds that move between the northern and southern hemisphere annually, and you can identify viruses, potential human infecting viruses in this guana and head that off. Another group, I actually have talked about this group at some length, is looking at mosquitoes in Brazil and what sort of viruses they're carrying, particularly the traditional arboviruses of dengue, chikungunya, Zika, but doing some sequencing for potential novel viruses as well. And then in Nigeria, we're working with a group that is looking at the co-infection of malaria and viruses in patients in a rural community. And what we want to highlight here is both the need to make all of these data systems local and the need to sort of respect data sovereignty and give people control over their own data. What we really feel throughout this is that the most important part of any global pandemic early warning system is taking advantage of the expertise and the knowledge that people have about their own regions. It is far better to have someone who understands the region, understands the systems, understands the disease dynamics of their country, think about how to measure disease in that area than it is to come in with a one-size-fits-all approach and to just try and force, one way of doing things. So while we're building the system, we're very focused on how we can work with local teams to do community-driven models that are going to fit both the local public health needs and provide local benefits, but also feed into a global predictive system that can build all of that stuff. The components of such an early warning system include identifying drivers, that's climate change, environmental disruptions, a lot of stuff that we've talked about here, building out data streams, structuring that data in a way that we can build effective prediction out of, and then providing these structured data, these structured drivers to these community-driven models so that we can give local people access to their data, access to a broad global suite of data, access to high-powered AI tools, compute, all that, and then power them to make some really effective outputs that are going to be very impactful for their local area, as well as global effective outputs. I said it earlier, and I'm going to say it again. Talent is global. Every region of the earth has people that are talented, people that are smart, people that want to make a difference for good. What is missing from any kind of global system is the opportunity to make things effective. And what we want to do with the pandemic early warning system is make that opportunity a little bit more evenly distributed, give people the opportunity to improve health and improve their understanding of disease in their local area, and then coordinate that so that effort is not just in one place, but that we can lift and modify and affect the same kind of change in multiple different areas. So what I want to close out here with is talking about the overall key takeaways. Genomics is a powerful, powerful data source. It's certainly going to be the basis of any pandemic early warning system, but it needs to be combined with these other data streams, particularly social determinants of health, climate data, epidemiological data, things that are going to give us this sort of big, robust 3D picture of data. And that what we need to do is both focus on making that data local and global. We need teams that can understand the local import of their data and teams that can understand global trends and global spread. Part of our work, we've been building out a pandemic early warning system and a platform for this called GeoSeek. If you're interested in doing this, please go ahead and go to geoseek.com, reach out. We also have a paper under peer review preprint right now that you can look at that writes up some of these ideas in a more in-depth way. So feel free to look at that, and we're always looking for people to collaborate with here and very interested in talking. With that, I'd like to just acknowledge the folks that have helped out. Of course, there are a lot of people at Biotia that have worked on this, but I want to highlight a few of the people that have done a lot of the work that you see in this presentation. In particular, Dr. Rafael Freitas and Dr. Gabriel Valu are leading the work that I showed in Brazil and building that out with field crews. And then many of the images and the satellite images you see here come from Descartes Labs and from Dr. Jim Golden, who's from Common Task AI, who have done a lot of work on how we look at this climate data. And finally, I want to highlight Dr. Charles Vorismarty and his group at the City University of New York, who have done a lot of work around climate modeling and weather modeling and how that can relate to health. So with that, I'm going to close out. Thank you very much for listening.